to start recording and then we're going to start the webinar. Good afternoon, everyone. Welcome to our Connections webinar. We'll let everyone join us. So we'll give everyone a couple minutes here. And welcome to the Our Connections webinar. Amy Garrettson, class of 2004 and Director of Constituent Engagement here from Smith Hall on campus, checking in with all of you. Kind of watching our participant numbers click up as we get started. While we're joining, if you all would like to put a quick note in the chat feature with your favorite Ripon College course, that would be an awesome thing to share as we get started today. Also, feel free to leave your name and class year, where you're messaging us or where you're joining us from today. Always fun to see who's on the webinar. Okay, we'll give everyone a few more seconds to join us. Again, welcome everyone. We have alumni and families joining us today as we start our fall Our Connection series. Welcome back, Zaya, recent grad joining us today. I just wanted to reiterate how important the summer Our Connection series was to the engagement at Ripon College, and our office really reached out and got a lot of participation from alumni across the country and the world in this webinar series. It was very productive. We're super happy to communicate with all of you in this way. Lots of familiar faces in the chat talking about their favorite courses, and we're excited to talk to you guys today. This fall, we decided that it would be a really fun way to kind of make everyone feel like they were back on campus for the fall semester by having a more academic focused webinar series. And so we're excited that Dean John Sisko is helping us moderate these fall sessions. He's new to Ripon, and we're excited that he's participating in this way. It'll give you all a chance to meet him since you can't come to campus and visit with him directly. And today we're also joined by Professor Mark Kainz from the biology department, who's going to kick things off for us as well. Again, we're going to have 3 more sessions this fall series with some faculty in some other areas, including physics, economics, and history. So stay tuned. Those details will be coming out soon. 2 more sessions in October and 1 in November. So we're super excited about that as well. And again, we're excited that both alumni and parents are together learning more about academics and campus life and that student function. Again, if you do have specific questions related to the webinar topic, please put those in the Q&A function, and I'll help monitor those and share those with the Dean and Dr. Kainz today. If you want to continue to chat with each other, of course, do that in the chat feature and keep commenting on the topic, of course. So at this time, I'm very pleased to introduce Dr. John Sisko, the new Vice President and Dean of Faculty. He's been with us an entire 8 weeks, which is good. The weather has been beautiful for the fall start, and we're happy to have him here on campus. Prior to coming to Ripon, I'm going to check my notes here. We don't want to miss anything up. John had served for 4 years as the Dean of the College of Arts and Sciences and the Wayland H. Cato Jr. School of Education at Queen's University in Charlotte, North Carolina. And prior to that position, John has a lot of teaching experience, over 20 years with faculty and administrative positions at Temple University, the College of William and Mary, and the College of New Jersey. So John is bringing quite a background to us. He is a professor of philosophy, specializing in ancient Greek philosophy, and he's an active researcher and continues to publish quite regularly. John received his bachelor's degree in liberal arts from St. John's College in Annapolis, Maryland, and his PhD in philosophy from Rutgers University. John says he came to Ripon because he believes in the transformational power of the liberal arts education, as a lot of us do. But he also confesses he is very grossly underprepared for the cold weather that is coming because he's been living in North Carolina. So feel free to offer him your best winter advice. He already told me he does not own a parka, so we'll have to work on that. And he's going to have to buy a snowblower. So, you know, things for John to be welcome to campus. His wife, Karen, is here with him in Ripon, and she's an archivist. And their daughter, Adriana, is completing her PhD in Women and Gender Studies at the University of Kentucky. So a bright family, and we're excited to have you in Ripon, John.
Thanks, Amy. Thanks for the introduction and welcome,
Bet.
welcome everyone. I'm really excited to be here at Ripon. It's a dream position for me. It's an odd time for everybody, but a dream position for me. So I'm really excited about that. It's a great liberal arts school with a fine tradition. So today I'm going to introduce Mark Kainz from the biology department. His wife Julia is from Peru, and she works for the Winnebago City Public Health Department in the battle against COVID-19. So that's very exciting. In fact, Mark himself last spring and summer was a contact tracer for Winnebago County as well. His daughter Sophia is an honors student in high school in Oshkosh at West High School. And she does equestrian and track and cross country as well. So Mark has a PhD from Cornell in molecular biology. He held a postdoc position at UW-Madison, taught at Colgate and Whitewater before he came to Ripon. And his research is on virus replication and virus particle assembly. So he's our virus guy. And he's been teaching in the Catalyst program, a program that he himself leads as the associate dean for Catalyst. That's our relatively new general education program that involves 5 courses that are thoughtfully structured around the sorts of skills that employers want to see in graduates, and graduate schools and professional schools want to see in graduates as well. So this is a snippet from a course that Mark is teaching right now. And without further ado, I'll turn it over to Dr. Kainz to tell us a little bit about pandemics.
Okay, well, thanks for the introduction. And just looking at the attendees, it's nice to see so many familiar names. Thanks for taking the time out of your day to join us. I hope I make it worth your while. For those of you who aren't really familiar with Catalyst, I want to tell you just a bit about it to put what I'm going to talk about in context. So as Dean Sisko said, it's a 5-course scaffolded series of courses that focuses on developing skills as opposed to disciplinary content. The idea is that we use disciplinary content as the vehicle to help students develop skills. And so there are 2 classes that students take in their first year, a writing class and a quantitative literacy class. As sophomores, they take a intercultural competence class and a class which I teach on integrative thinking and collaboration. And then this culminates in their junior year with the Applied Innovation Seminar. And so the idea is all of these classes build towards them succeeding in, the Applied Innovation Seminar. So the course that I teach is called Bring Out Your Dead, Infectious Disease Through History. And it's a course that I developed actually with Becky Matsky. Since then, we taught it once together. And then when she had to take on other administrative duties, I got it all myself. Fortunately for me, I had a really good person to learn from in Becky. And I also was really interested in history and took a lot of history in college as an undergrad. So with that as prelude, let me talk about one of the topics that we talk about in Bring Out Your Dead. And so the idea here is, in this part, is integrative thinking, how we would address a question or a problem looking through the lenses of 2 different intellectual approaches, in my case, biology, in Becky Matsky's case, history. And so with that, let me go ahead and share my screen. And what this actually is going to be a bit of an opening lecture that I give. And the idea here is these are sophomores. These are sophomores who, many of which don't have any background in biology or the sciences. And so the idea is to give them enough science so they can actually make some thoughtful choices when they're starting to work through the history part of it. Okay, so the topic for today is the 1918 flu. And so to get started, there are 3 strains of influenza called A, B, and C. Influenza A is the so-called pandemic strain. It's the one that we worry about a lot in terms of global pandemics. Influenza B is no fun to have if you're infected with it, but it's not a pandemic strain because it basically only infects humans. And as we talk about how these pandemic strains develop, it'll become a little more obvious why that is. And then influenza C is not a particular significant health threat to people. Okay, so I want to say a little bit about the virus, and just a little bit. So if we think about the virus, there are 2 important proteins called hemagglutinin and neuraminidase. These are the H and the N in H1N1 or H3N2, those kinds of things. And they actually have 2 roles that are important from the point of view of people. One, they are important in how the virus can get into a cell and get out of a cell. And also because they're on the surface of the virus particle, that's the primary target that our immune system sees. So when you are infected with the flu or you get the flu shot, most of your immune system is directed at those 2 proteins. Okay, and then another thing that is important is that its genome is RNA as opposed to DNA like we have. And that's important for reasons that I'll tell you in just a second. And then finally, each gene that the virus has is on a separate RNA molecule. Think of them as chromosome-like. And the fact that each gene is on its own separate RNA molecule also is important in the generation of pandemic strains. Okay, so here's a little cartoon of influenza. These little lollipop-looking things on the outside are illustrations of the hemagglutinin protein and the neuraminidase protein. And then inside are the various RNA molecules that make up the genome. And what I want you to notice is that each one of them encodes a particular viral protein. Okay, and so here's basically from the point of view of the virus, what has to happen for it to be successful, right? We have to have an interaction between the hemagglutinin protein and a sialic acid receptor on the surface of the cell. The virus is internalized, and then the virus escapes the endosome and starts its foul replication stuff that causes us disease. Okay, so if we think about this, right, what has to happen for disease to occur is the virus has to be able to get into the cell and it has to be able to get out of the cell. And the hemagglutinin and neuraminidase proteins are what are the 2 primary things that govern that. And I'll circle back to this in just a second. But the other thing that has to happen is the virus has to be able to replicate or multiply in cells. So if it can't replicate, it cannot cause disease. Okay, and so, and this is important in terms of what we call virus adaptation, which again, I'll circle back to. So the virus encodes a number of proteins that are involved in replication. And one that is really important is the so-called replicase. It's the thing that copies chromosome from the entering chromosome to copies. And the reason it's important is that it makes lots and lots and lots of mistakes. If we think about our own cell replication, the replication of our genome makes a mistake about 1 in every billion bases. Influenza makes a mistake about 1 every 100 bases. So that gives you an idea of how error prone this is. And then the other thing that has to happen is the virus actually hijacks normal cell proteins for viral functions. And if there's not a good match between the viral proteins and the host cell proteins, then replication doesn't occur very well. And again, that's part of adaptation. Okay, so as I said, influenza A has a number of hosts, and on this diagram I've tried to illustrate them. What I want you to notice is that the key host here is waterfowl, wild waterfowl, ducks and geese. And then the dotted lines indicate possible transmission from that species to another species, although not particularly efficient transmission. And the solid line indicates pretty efficient transmission. So what I want you to notice here is that the duck then is the central player in this, and it encodes, duck viruses can infect basically just about everybody else, and that's important for maintaining the virus on the planet, and also for generating new strains. Okay, and then in this diagram that I stole from one of my lectures, if you remember the 1997 Hong Kong flu, that was a direct transfer from chickens to people. And that's one of the reasons it was such a serious disease at that time. Okay, so keeping on the idea of host range, right? The tissues in which the virus targets are different in the different hosts. So in waterfowl, it's an intestinal virus. And it doesn't really cause any significant disease symptoms. The ducks are constantly depositing it in the water and becoming reinfected. So you may wonder, if you go swimming in a pond that's got ducks on it, why don't you get the flu? It's the wrong strain. It doesn't readily affect you. So if you're swimming in a pond with lots of ducks in it and you get water in your mouth, you're getting a ton of influenza virus particles, and they're not doing you any harm because they can't infect you. The rest of us all suffer a disease that is respiratory. Okay, and so this is one of the primary drivers for why influenza is a serious pathogen for these kinds of animals, not so much for waterfowl. Okay, and so if we think about transmission, with the waterfowl, it's an oral fecal transmission, right? They excrete the virus, they reacquire it. With the rest of it, it's primarily aerosols. And those can be in two ways. One, someone coughs or sneezes, makes a big aerosol, somebody else breathes it in. That's an important mechanism of transmission. The other is someone coughs in his or her hand, touches a doorknob, shakes your hand, whatever. Because the virus is highly stable, that means it can really cause a problem. Covering your mouth with your hand is not a good idea, and that's why hand washing is recommended so often for controlling the virus. Okay, so this is an important point here. The virus is constantly changing, and it changes in two different ways. So recall how I talked about how the replicase makes lots and lots of mistakes. This is what causes what is called antigenic drift. And what this means is, over the course of a year, lots of little changes occur in the hemagglutinin protein. And what the result of that is that it is still able to interact with the receptor on our cells, but it changes bit by bit by bit, so that the infection that you had last year or the flu shot that you got is not very effective anymore, because the antibodies you raised don't recognize this new strain of the virus or this new version of the virus very effectively. Okay, so that's that part. Where pandemics start is what we call antigenic shift. And in that case, we have a mixing of viral chromosomes between different strains of flu that generate a completely novel version of the virus, and one that our immune system has not seen. And so here's, I think, yeah, here's an example. So from the point of view of people, pigs and ducks are the main players in this, and pigs in particular. And so on this diagram, what I want you to notice is, here's the pig. Here is a strain of virus that infects people, and one from some other species, let's say duck. If the pig is infected by both strains and we undergo viral replication, if they happen to be in the same cell, right, you can end up with some virus particles that look just like the one that came in and some that look just like this, but you also get ones that are a mix of the two. And this is the version of the virus that causes pandemics. Okay, this is one of the things that was driving the 1918 flu. Okay, so then what has to happen? The virus, as I said, has to be able to replicate to be transmittable. And this often requires adaptation. Okay, so for example, if you remember in 2008, when people were really worried about the bird flu, that was an H5N1. Okay. If you were around lots and lots of chickens, you could become infected. It was a very, very serious disease. It could be fatal. But from the point of view of public health, it wasn't very easily transmittable from person to person because it wasn't able to replicate in the cells that line your trachea. And so when you cough, you weren't able to release particles. And that's why we didn't have person-to-person transmission. Although if you were unfortunate enough to get it, you were not in very good shape. So one of the issues that people were very concerned about was, well, we're a few mutations away from the bird flu adapting to people, and then we've got a big problem. Well, then came 2008 or 2009, I forget, H1N1 showed up, everybody forgot about the bird flu. It's still out there. We can still worry about it if we want. Okay, so now let's talk specifically about 1918. So this was a worldwide pandemic, and it was a situation that you can almost call a perfect storm, right? We had a brand new virus, the so-called H1N1. This had not been in circulation before. So it was a novel virus worldwide. Where it came from, we used to think we understood quite well, and now we don't understand nearly as well as we thought. There is really good evidence that shows that there was an H1N1 strain in a swine farm in Kansas, and it moved from that farm into military bases, and from there it went all over the place. And this was kind of the party line for a long time. But then some recent data has emerged that shows that construction workers that migrated from China to North America were suffering from a flu-like disease that was remarkably similar to H1N1. And so there's a lot of forensics being applied to trying to figure that out. Regardless of that, the thing that was important is what we now know from studying H1N1, the 1918 version, is that it was a mixture of genomes from swine strains, waterfowl strains, and human strains. And so this meant that our immune system was completely naive to it. We had basically zero protection. Okay, and then what else was going on? World War I. And this was one of the things that contributed to kind of the so-called perfect storm, right? We had living conditions in terms of the civilian population in the United States, even more so in Europe. We had living conditions of the armies on both sides of the conflict. We had mass movements of people associated with the war effort. And we had media censorship, so there was not a lot of information getting out. And one of the reasons it's called the Spanish flu is that Spain wasn't particularly involved in World War I, and so they weren't censoring their newspapers, and that's where it got reported, and so it was called the Spanish flu. Spain was not a particularly big hotspot for the virus, but that's how it got the name. Okay, so what are some other things that are important about the 1918 flu? Well, one thing that was very unusual is it was really lethal in young adults. Now, usually influenza is very lethal in the very young and the elderly because their immune systems are not very developed yet with the youngsters, or their immune systems are not what they used to be in the older folks. But people in their 20s and 30s are at the prime of their life. Their immune systems are quite strong and usually are able to fend off lots of infections that people on the other ends of the age spectrums can't. In this case, these individuals were at particular risk of serious consequences, and the contributor there was the strength of their immune system. So there are signaling molecules in your immune system called cytokines that signal between different cells in your immune system. And what can happen is if your immune system gets hyperstimulated, it generates what is called a cytokine storm. And the results of your immune system and your immune response is what contributes to the severity of the disease symptoms. So with people of that age group, they had robust immune systems, they made this massive cytokine storm, and they suffered because of it. There is interesting conversations about the effect of influenza in 1918 on World War I. There are some historians who say that it was a major contributor to the end of World War I. And this is a really cool hypothesis. It's really appealing. But there's other historians that say it was a problem, but there were other things going on that brought about the end of World War I. I'll let the experts in world history address that more fully. One thing that we do know was that on both sides of the conflict, the German side and the Allied side, there was a really difficult time maintaining troop strength. And we do know that the Germans were planning a massive offensive that they thought was going to break through, but they couldn't deliver enough troops to the front to actually let it happen, and that tended to bog things down. Now, the other thing to think about is the effect on society. Okay, and some of these things may seem a little familiar, right? We had a big damage to social fabric, okay? So why? Well, perhaps sounding familiar, there was resistance in different parts of the country to wearing masks. Wearing masks was effective in helping control the spread. It wasn't a silver bullet, but it was effective. And also suppressing or limiting public gatherings. There's lots of nice examples where a city decided to have a parade, they had the parade, and then they had a big outbreak of 1918 flu. Other cities that did not. We had decreased cooperation. And, you know, in the United States, there's a big history of neighbors helping neighbors and communities working within each other. One of the problems then was the fear of becoming infected made people less likely to help their neighbor. People were much more on their own. And then again, something that might seem a bit familiar is the healthcare workers started to just become exhausted. And in particular, skilled nursing was a big problem. Skilled nurses were by far the most effective group of people in terms of controlling the 1918 flu. Why? Because they were good at kind of the public health point of view. Remember, in 1918, viruses were known, but they weren't widely understood. And the understanding at the time was that flu was caused by a bacterium. And so there was a lot of effort put into identifying the bacterium that caused the disease. The nurses weren't concerned about that. The nurses were concerned about preventing transmission and getting people well. And the nurses were remarkably effective. There are some historical sources that show that a single nurse showing up in an isolated community basically turned the health of the community completely around, which I found amazing. And then the last thing I'll talk about and then open it up for discussion is because of the effect of flu on the young adults, we had, and in addition to the effect of World War I, we had a big depopulation of people in that age group, in particular males in that age group, because they both suffered from the flu and they were soldiers. And so what this did then was it gave us kind of a loss of people who are generally the drivers of our society and the drivers of our economy. Those young adults are the ones that tend to really push economic development and societal change. And so that had an ongoing effect that lingered long after 1918 flu was just a bad memory. So I think I will stop there, and I'll be happy to talk about whatever people want to talk about.
Well, thanks, Mark. That was a fascinating overview of just one snippet of one day of class here at Ripon. And I'm sure at least many of us, certainly myself, hadn't understood how the moniker Spanish flu was attached to this bit of influenza. So that's very interesting. I have a few questions, but I want to go to the questions first that alumni and parents have submitted and related, but many of them are more focused on the current pandemic.
Sure.
So Ashley Skaczynski, class of 2009, would like to know why Wisconsin has some of the highest COVID rates.
That's a really good question. We actually, to put not too fine of a point on it, Winnebago County is one of the poster boys for high rates of transmission. We actually have the CDC in visiting Winnebago County right now to try and figure out why. One contributor, although it is not the sole contributor, was the way we came out of lockdown. So Wisconsin, like many other states, was in a pretty strong lockdown to try and prevent spread of the disease. It was pretty effective, and then there were obvious economic costs to that, and so the idea was, well, let's open back up. So one of the issues was, how do we open back up? The governor proposed a plan. The legislature didn't like it. It went to the court. The plan was thrown out. In my view, one of the big mistakes was, instead of going back and revising the plan, it was just, there's no plan. So now everyone did what they thought was best. Perhaps they did what they thought was best for themselves, for their business, or whatever was driving them. And that was, I think, the thing that kicked this off, and one of the things that is keeping Wisconsin kind of in the news.
Thanks, Mark.
I'm sure there are plenty of other
question
things.
From Liz Walsh, class of 14. This is a very philosophical question. The question is really, knowing that certain theories, scientific theories in the past, have been debunked. When we look back in 100 or 200 years, what sorts of theories do you think will have been proven to be false? That's a big one.
Well, you know, Liz is actually going to be giving a Zoom lecture to the biology senior seminar in a few weeks. And I'm going to let her answer that question in a couple of weeks. Because
Great.
truthfully, I have no idea.
Right. No, that's very good.
Our scientific understanding now is quite advanced to what the kinds of things that people thought about bad air and miasmas and stuff of the past.
Sure.
So I'm sure that we've got plenty of ideas that are completely wrong, but they're hard to see from here.
Well, I could actually speak to the humoral system. The black bile and yellow bile as a specialist in Aristotle. Aristotle developed that theory with no empirical evidence whatsoever.
Mm-hmm.
And then because Aristotle had this great systematic philosophy, he influenced thinkers to like the 1600s, Galen being one of them. So I would say the one difference is there was actually no empirical evidence for that one, right? So
Right.
we're in better shape now. At least we're wrestling with empirical evidence as we look at different theories. I don't know if you
Right.
think that's correct, Mark, but I'll throw it out there.
Oh, for sure.
Okay. This is a good one from Peter Kolowski, class of 72. Did the federal government take a lead role in trying to contain the 1918 influenza? And I think there's an implicit question of comparison.
Yeah, I sounds about right. The government did have a role, but in 1918, we did not have the Centers for Disease Control. We did not have the National Institutes of Health. None of those big government agencies. And the federal government was pretty busy with World War I. However, there were lots of government-funded scientists who were working on it, working on the flu. So our system was actually in 1918 kind of similar to what we have right now, where we had states and localities that were largely governing what the response was. They were talking to each other, but in the face of a huge nationwide pandemic, it's far more effective to have one plan, one way forward, as opposed to 50 kind of similar ways forward. And I think that was a contributor to the problems in 1918. And I think it's a contributor to our response now. If you look at some of the countries in Europe or South Korea, where they have a national policy, they're being far more effective at controlling COVID than we are.
Great, thank you.
I think I was going to say
good
there's a
question.
couple questions in the Q&A I think fit in timely with this, and there's a little bit of overlap between several of them, but you talked a lot about obviously the 1918 pandemic, and so what can folks today take away from that, and do you think there's anything we can learn or should have learned that we can apply to today? And then also, do you think that the effects of COVID on our society will be similar to those of the influenza in 1918?
Great questions.
Oh, that's a fantastic question. Yes, we could have learned a lot from 1918 in terms of having a unified response, trying to get out ahead of it. To digress for just a bit, if you remember the swine flu in 1976, people thought that there was a soldier at Fort Dix that died of influenza. They looked at it, it was H1N1. They thought this could be 1918 all over. And what was it? The Department of whatever the precursor of Health and Human Services was, mounted this huge vaccination campaign at a time when flu shots were not common. And if you remember, President Ford even was on TV getting a flu shot to show everyone that it was okay and stuff like that. And the 1918 flu, H1N1, never really showed up. And so there was a lot of finger pointing, and public health people said, that's great that it didn't show up. We did the right thing. If we had gotten behind on this thing, it would have been a disaster. And I think that's a lesson that we should have learned from 1918. that would have been helpful now. You know, on the other hand, there's a limited amount of resources that the country has. And so it is hard to make a case for, we need to have a lot of money devoted to infectious disease when there are other problems that are causing problems right now, as opposed to the threat of infectious disease. So there needs to be some sort of middle ground or nimbleness or something that needs to change. But yeah, I don't think there's, you know, if you want to look at COVID and point fingers, there's plenty of places to point fingers throughout our society and government. I'll leave it at that.
I had a question from Noah Lee, class of 2004. And to paraphrase the question, I think the question is really another thought experiment. Like, if there were no infectious disease and there hadn't ever been infectious disease, what would be different about the world now compared to the way it is?
Wow. If there were no infectious disease, well, we probably wouldn't be here because the world would have overpopulated like a long time ago. And we would have exhausted all the resources. You know, one of the things that we've seen with infectious disease is we conquer something and something steps up to take its place. You know, people used to die of all these infectious diseases, and then we got antibiotics, we got vaccines, and then what happened? People started dying of cancer and heart disease and stuff like that. So something's always going to kill us. To use my typical optimistic way of looking at things. So yeah, I think infectious disease is always going to be around. If there never had been infectious disease, wow, what would be interesting to think about?
Wouldn't that be a great seminar discussion with students? Like, what would have happened? What could it be like? It's a
Sure.
phenomenon.
And what would you
No one
have
can
gone to
come talk
graduate
about
school?
that.
Yeah, good point.
And what would you have gone to graduate school for, Mark, if you didn't have a problem with viruses?
Very good. Maybe one more question as we kind of are getting close to the end of time today.
Sure. I think
Favorite
that,
one, John? What did you see that was?
Great. I think there's a general sort of ballpark question. It's like, if you were going to take one key lesson away from your study of the 1918 flu and its relation to COVID, what would that one key lesson be?
A uniform, sustained response is what is needed. Nothing more, nothing less. If we have that, we're going to get ahead of it. We're going to get on top of it. If we don't, we're going to be flailing for quite some time.
Thank you for that optimistic closing, Mark.
My students know that. Yeah.
Good. Well, thank you both so much for taking the time today to talk to us. I think it's fascinating. We could have a whole series of just talks about the 1918 flu pandemic and what does that mean for us today. And hopefully we can continue this discussion, Mark. And Dean Sisko, thanks for joining us. And we welcome you to the Ripon community and excited to have you as well. As a reminder, the webinar is being recorded and we'll post it to the college's YouTube channel. After, I will also send a note to all of our participants with a little short survey and some follow-up. And I'm sure Dr. Kainz would be happy to hear from anyone with specific questions. And so if you'd like to get in touch, we'll include that in the email as well. Definitely stay tuned for more information about our next web series. I can say on October 13th, Dr. Chris Othon from the physics department is going to join us to talk about her Catalyst course called Life and Death in the Universe. And so we'll look forward to hearing about that from her and a little more discussion there. And then again, watch for some more sessions coming up. So I hope you all have a great day. And again, thanks for participating.
Yes, thanks everyone. It's been nice to see some familiar faces or familiar names.
That's right.
Thank you,
Have a
everyone.
good day, everyone.
Take care. Bye, everyone.
There we go. People are ticking off pretty quick, so that's great. Nice commentary. All right. Well, have a good day, John. Nice to see you.
Okay, Amy. Thanks so much.
Take care.